The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 18th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcast, The Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian. I've been in the flow cytometry industry as a CRO for over 13 years.
Thank you, Brian. And we're joined today with three special guests. Barry, would you like to introduce yourself?
Hi, I'm Barry Morse. I work with a consulting group called the Therapeutic Innovations Group. Fan of the podcast and been a fan of Adam's for even longer. So, we worked together for a long time, and I'm really happy to be here with you guys today.
Thanks, Barry. Hillary?
My name is Hillary Quinn, and I've been working with Barry and Mike for several years now, and right now in the Therapeutic Innovations Group. And my background is in vivo pharmacology. Happy to be here.
Thank you, Hillary. And last but certainly not least, Mike.
Thanks, Adam. I've had the pleasure of working with Barry and Hillary for decades. Spectacular. And years and years working with Adam, unbelievable human being who, got to know, we grew up together, and that was spectacular. And, had the pleasure of seeing a lot of drugs move into the market. And so a lot of experience there, and looking forward to moving more products into the clinic as well.
Thanks, Mike. I'm looking forward to our conversation. Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with additional location in Europe and a partner location in Australia, we're both here in Philadelphia, and our producer, Jeremy, is in Missouri, so we're truly all over the map. We're exhilarated to have you listening to the 18th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're ecstatic to have you here, and we're looking forward to a fun episode today. To kick us off for episode 18, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. I'm particularly proud of this agenda for this podcast because we were able to get it quite a bit in advance of the actual episode recording here. So always a good thing.
Yeah, it really is. And I appreciate the effort there. It's always good to have a little bit of time before we do the podcast.
Indeed. And in our first bit of medical research news, I like this one for its title because it's one of these sort of breathless titles that sounds really exciting. Scientists discover a new part of the immune system, like something had just been hanging out and we just hadn't seen it for several decades, right?
Yeah, I'm really curious where breathless comes into that, but I know that's a common statement. However, to your point about the immune system in here, what we're looking at are proteasomes, right? And it's known that proteasomes break down proteins specifically in relation to antigen presentation for specifically T cells to be able to drive the adaptive immune response as a kind of corollary to antibodies that recognize more of a native full epitope T cells recognize broken down bits of protein on the surface here. And so that's the primary role that everyone is aware of for proteasomes. However, here, this paper looks at an alternative role in that breaking down proteins will actually yield antimicrobial peptides, which is very unique. And so was interested to see how they went about doing this. And so curious, Brian, on your thoughts here.
That's exactly it. And, the team used something that they called dumpster diving for this to look at bacteria and how these small bits of peptides impacted them. So yeah, it was a very unique paper. It did. So I provided the link to the BBC paper or article that just described the paper, but it does reference a full Nature article. So there's some actual science behind it. And yeah, I second your opinion. It was very novel how they went about finding it, and interesting that they're able to look in a very well studied place and come up with some interesting drug targets here, basically.
Yeah, and artificially digesting the human proteome, right? The entire proteome, coming up with 34 million peptides. And then from that, one of the things they were looking for were cationic termini. And so that would indicate something that could potentially interrupt the bacterial cell wall. And they came up with roughly 270,000 of these peptides, tested them against different bacterial species, and did see titratable activity there. So really interesting. Could be an alternative here to the general antibiotics that we're used to.
Indeed. And, I think anything that leads to more of these antibiotics, I know it's not a terribly profitable or large part of the pharmaceutical industry, but, it is a general public health concern. So the more options we have, the better and the safer, all of us are. And ideally, we can get rid of some of these long-time bacterial diseases at some point as well.
Well, especially too, if you're talking about antibiotic resistant strains, right? You need to have more tools in the toolbox to be able to counter that. And so this could just be one of those tools.
That's it. Yeah. Yeah. So exciting article there on immune system and some of the surprises that it still holds for us. And then the next one is potentially not really a medical research news because it has economics in it, but I thought it was interesting anyway. Just the lifetime health effects and cost effectiveness of tirzepatide and semaglutide.
Right. So these are the GLP-1 drugs looking at weight loss specifically. At least there's a big market for them in weight loss, essentially increase insulin release from the pancreas. So regulating blood sugar, decreasing appetite by slowing gastric emptying. And so these drugs really have a wide range of use, and especially with the large prevalence of obesity. These drugs can help and reduce percentages there and overall make the population healthier. And so what they were looking at in this paper was, is that still something that's cost effective, right? And so they had to take a look at quality adjusted life years, setting a benchmark, and then determining if they are profitable. And the interesting thing were a couple of the drugs needed significant reductions in cost to actually be profitable. One was 30.5% reduction in cost, so from 6,000 to 4,000, and the other one, 81.9% reduction in cost to be profitable. Now, obviously, take a step back and say this is looking at it probably more likely through the lens of healthcare, health insurance, things of that nature, evaluation. But obviously, the patients are going to argue that that cost effectiveness, really, you need the medication to increase health. They did compare it to lifestyle changes and this plus lifestyle changes. So realistically, looking at making it sustainable. But it was really cool how they built the models to be able to evaluate this sort of thing. And it did have the caveat that some of the assumptions may not necessarily hold, and other evaluations had different numbers. But it's important to understand, frankly, I mean, you're talking about people's lives, and the cost of healthcare is quite significant in many areas.
That's exactly it. And what I found interesting about this paper, and what I don't think about on a regular basis, at least in the CRO model, is, if I have $1 to spend on healthcare, where will I put that dollar to, improve human health the most? In this case, they explored, is that dollar well placed with an obesity drug like semaglutide or any of the GLP-1s? And as you say, the answer in some cases was yes, some cases no, based on the current cost. But it's right on the cusp, and that means that probably someone in the pharmaceutical industry is quite good at estimating that, right? Because if it was really far one way or the other, then the answer might be no, and they don't want that. So yeah, so I feel like that is interesting. And then also always interesting to see what these calculations are. This, you can think of it as a relatively healthy population if you're talking about the obese population. But we usually think of course, of cancer and oncology indications that, this calculation is significantly different there as well then. So good to think about it from there. And especially as we think about advanced therapies and how they're put through similar frameworks to see if a drug is worthwhile from an insurance perspective.
Yeah, absolutely.
All right, and then on to the actual investment news. I lined up the first story as a new early stage venture capital firm. So Sofinova Partners is generating a fund of about 165 million euros with partnership from Big Pharma to build early stage biotechs in the EU.
Now, I'm going to be completely transparent here. This is interesting. I think it's certainly helpful. I'm going to be interested in your take on it and your inclusion here. So just some of the background on it. It's the second such fund, 10 times the first fund as far as value is concerned. And we're talking about, again, Europe, like you mentioned, specifically Italy, France, UK, and Denmark, looking at early stage development acceleration. Now, two of the things that they had mentioned, why this fund, what does this do? So they're looking to be able to seed where companies need support for anywhere from 2 to 5 experiments that are critical to get to Series A funding, right? And so what's Series A funding? That's the first round of venture capital funding as for a private company. And it comes after the angel or seed investments. What's significant about it is the company's demonstrating their ability to earn money. And so that's why these rounds of funding can be rather large, but also very important for company health. Now, this fund expects to be able to help 5 to 8 companies that would come out of this fund. I guess the way they put it is they'd be legit companies or whatnot. But yeah, curious as to your addition here.
I thought it was very interesting to see how a startup basically transitions from usually academic or institutes or driven research, so like the medical research that we just read about, into a functional company. And there's, as you say, a lot of money involved in making that leap. So this fund is there to basically establish those proof of concept experiments, if you will. And then for the ones that make it through that sort of kicking the tires exercise, then they will fund the Series A to then ideally get them all the way to a clinic or somewhere that a biotech or a pharma company can pick them up.
Now, I'll be very curious with our guests on the show, some of their experience in this area, if we do get the chance to discuss a bit, because, in some of the experience that I've seen, it's in large part decentralized as far as getting funding, and you have to go out and you have to meet with a lot of venture capitalists. The different rounds can be challenging, but I've never gone through it myself, just heard secondhand. So it's always interesting perspective to get.
It is. And it's interesting, too, that a lot of the people contributing or people or entities contributing to this fund are big pharma companies themselves, right? We've talked on a couple episodes about how a lot of innovation is sort of being separated from the main portion of the larger companies, and the R&D is being done under those smaller organizations. so that if one of those directions doesn't work out, then it doesn't sort of drive down the bottom line of the larger company there. And that seems to be the case here. And as we go into our next story, you can see what happens at the end of this. If all the experiments work out well and the drug performs as hoped or expected, then ideally all these companies can work towards an exit with one of these pharma companies that's funding the fund, which is interesting.
Yeah, I'm really excited to see this next story. So a perfect segue there. I'll let you tee that one off.
Sure. So here, the next story is on AstraZeneca, and they've purchased a new Belgian company that's and it's a $1 billion deal. But this Belgian company called ESO Biotech is actually making CAR T cells inside people, as opposed to treating the cells ex vivo and then sort of infusing them. Here, they're using a lentivirus to modify cells inside people.
Yeah, and so we've talked about cell therapy before, and when you think of off the shelf cell therapy, I can say this. I know I can. You're thinking about allogeneic cells, essentially generating the cells, freezing them down, and then you can put them into the patient, ready for use, kind of thing. But there's a lot of hurdles to be able to get to that point. A lot of cell engineering that has to happen to make it so that the body isn't rejecting these cells, since these cells aren't coming from that specific patient, which to what you were referring to, the autologous cell therapy, where you would be taking the patient's cells, modifying them, putting them back in the body. And so here, again, it's off the shelf. These engineered nanobody lentiviral particles will essentially go to the T cells and basically generate so that they would kill the cancer. One of the important things about lentivirus is they infect dividing and non-dividing cells, and so very effective at getting the DNA payload to where it needs to go. And here, since you're adding the lentivirus, the nanobodies in there, you are getting around some of the immune recognition issues that you would have for an off-the-shelf allogeneic platform. And so it's a way of circumventing that and making it feasible in a different scale, right? So you're making the virus and then you're inserting it, making the nanobody lentiviral, however you want to refer to it, and getting that infection. So yes, obviously demonstrated success. I mean, you don't purchase a company for 1.2 billion with no demonstration of success there. Really excited to see where this goes.
Absolutely. As you say, one of the biggest challenges for cell therapy is really scaling it, right? And as you say, allogeneic is a good way to do that, and then this could be another way. Manufacturing viruses is no small feat in itself. So, still going the lentiviral route, it's got its challenges, but it's likely easier to get significant quantities of a single lentiviral vector than it is to get quantities of either many different types of cells or cells from many different patients. So an interesting and economically, at least, interesting and feasible approach there.
Yeah, made lentivirus before for generating stable cell lines. However, did go on the site, was trying to look for more information around the nanobody aspect of it. Didn't get too much, but certainly something that I want to continue to look into further.
Sure. Yeah. Excellent. And then the last piece of news that I want to advertise before going into the main portion of our talk today is just all of the poster presentations that have been accepted at upcoming conferences.
Yeah, so we've got a couple of conferences coming up. We've got a poster at WRIB. That'll be our David Ambrose going down again. He had raved about WRIB when he went down a couple of years ago and couldn't wait to get back. So glad he's able to do that. And then myself and Dmitry Nigorev of our team, one of the scientists, will be going to Cyto, which is in Denver. I haven't been to Denver since I think I was a teenager. Back then, I was going for hiking, not poster presentations. I think it might be a little bit of a different experience for me, but really looking forward to getting out to Cyto.
Nice. Absolutely. Yeah, and we'll be talking about multi-site instrument standardization, and then separate, but in some ways related, the neutrophil oxidative burst assays. So exciting times there.
Mm-hmm.
All right. Yeah, and with that, then we'll sort of jump into the main section of our talk. I did select this paper from the Frontiers of Immunology called Monitoring of Circulating CAR T Cells, Validation of a Flow Assay for Cellular Kinetics and Phenotype Analysis following... I'm not even going to try this name.
Tisagenlecleucel.
Okay.
So Kymria, sorry.
Right. Indeed. Yes.
Yeah. So do you want to guess the impact factor?
I will guess an impact factor for the Frontiers of Immunology of 4.
5.62, not bad. Yeah.
Mm-hmm.
And so this treatment that they're looking at here was actually the first approved CAR T cell therapy developed in conjunction with the Perelman School of Medicine at UPenn, Novartis there. And so what the paper's looking at is monitoring medium and long-term persistence, pointing out that it's key, looking at peak expansion and trying to then correlate successful treatment, right? And they're using two different platforms here. The paper specifically is focusing on flow cytometry, but they did then correlate real-time PCR data with the flow data.
Okay. Excellent. Maybe this is a good time to introduce our guests in a little bit more detail. So for the Therapeutic Innovations Group, can I ask you to give a little bit more backstory on why you're here today and what your backgrounds are?
Why don't we go in alphabetical order? So we'll start with Barry.
Yeah, glad to join you guys today. You know, I've known Adam for a long time, you know, and I've listened to a number of your podcasts. You know, we've started our own podcast, and you guys were helpful enough to kind of give us some hints on getting that started. Yeah, so I think my background, you know, when I was a younger guy, I was a virologist coming out of grad school. But early in my career, I kind of started working at a company called Centocor, working on therapeutic antibodies. They had just been bought by J&J and kind of were absorbed up into the mothership. That's where I first met Adam. And worked for about 17 years there on therapeutic antibody development. My career kind of spanned a little bit. I was hired into the development group working on manufacturing cell lines, and then transferred up more into the discovery arm of the organization, working on therapeutic leads and led a protein expression group that was responsible for expressing these early leads and having them screened. After J&J, I was lucky enough to join a cell therapy group, Century Therapeutics. I think I was employee number 8 there. You know, Mike, who's also joining us today, you know, brought me in. Really a great experience, you know, with the startup, you know, building out that organization, you know, I was head of the iPSC biology group there. And, you know, it was so much fun to sort of, you know, be unloading things from the dock, you know, in the lab, you know, but also in high level discussions and building out the processes that resulted in the first Century Therapeutics product to go into patients. So that was a great time. You know, it was time for a new thing in last year in October, you know, and we at that point in time decided to start our group for consulting. So we started the Therapeutic Innovations Group. Maybe I'll let Mike talk about that a little bit more. but we are a group that is out there to help people who are trying to build processes to are just getting started. Particularly, I think our expertise is in companies who are trying to get going. Academic groups who are interested in starting a small biotech. We know how that works, and we're here to help you.
Excellent. Thank you, Barry. That was a great intro. You make it out like, we started working together a long time. It was, what, 20 years ago? I mean, that's
Yeah,
not that long ago.
you are a young pup.
I was very much a young pup. I was still in my 20s
I
at that
was
time.
a semi young pup, not as young, but
And you can imagine the horror. Like, I was told I was going to be reporting to you, and here I'm a Yankee fan and you're a Red Sox fan.
yeah,
But somehow we made it work.
you made it work. Yeah, it only got tense a couple times, I think.
It did. Well, that's why I had my Yankees thing right on the top of my cube, just so you could see it every time.
Every day, every darn day.
Excellent. All right, Hillary, would you like to give an intro?
Yeah, sure. I'm still a young pup, so sorry for you guys. Now, yeah, so like Barry, I started my career at Centocor just after their acquisition by J&J. I started my career and kind of have continued in this direction with preclinical oncology, mainly pharmacology. So, functional assays, whether in vitro or in vivo, mainly in vivo, but modeling and everything, starting out at J&J with large molecules and biologics, moving into small molecules. My family and I were sent on an expat, or I was sent on an expat assignment, and they just happened to come with me for 2 years. And we lived in Belgium, and Belgium is where J&J develops its small molecules. So for 2 years, I got to lead the in vivo group in Belgium, and that was a great experience. I learned everything that I know about small molecules there, which isn't a whole lot, but it's more than I had before that. And then I came back to Springhouse, or to the U.S. side, in 2017, and jumped back into large molecules. But then we started transitioning to autologous cell therapy. So started kind of dipping my toes into that. And in 2019, the opportunity came from Mike to join Century Therapeutics, and it was a great opportunity. It was so exciting. I joined as the head of pharmacology there, and after several years had built up a pretty decent size group, and we ran all the oncology in vivo modeling from discovery and screening all the way through IND enabling studies for our clinical candidate Century 101. So it was a wonderful experience. I loved that startup environment, getting to build a team and getting to work with everybody to get a company going, and wearing a lot of hats. And yeah, in October, I think the company changed direction a bit. So Barry and I and Mike decided to consult together with the Therapeutic Innovations Group. And then recently, I've had to step back from that because I just joined LabCorp. For those of you that don't know, LabCorp has a drug development group, and they do contract research. So I'm a director in the preclinical oncology or oncology discovery group for scientific development. So that's kind of where I stand, and that's all of my experience.
That's really a cool journey. I'm curious, have you been back to Belgium since coming back to the States?
We went in 2018, so a year after we came back. We went for 5 days to visit our friends, and because we missed it, and then coupled that with like a trip to Ireland as well. But that's the only time. But we do miss it very much. We liked it so much that I actually interviewed for and got a job as a head of discovery for Crown Bio in the UK. And our goal was to stay in Europe. But then kind of right before I accepted the position, my family and I all talked, and I think everybody but me was really, really homesick. So I decided against it, and we came back. But yeah, we love Belgium. I would love to go back sometime soon.
Very cool. And Brian, you'd been to Belgium not too long ago, right?
I've never actually been inside Belgium. I've been to France and the Netherlands, so all around it, but never actually in Belgium.
Okay.
It's a small country, and people forget about it. I think they've had between the Netherlands and France, they've been attacked from all sides in Germany. So, yeah, but it's a great little country.
Well, speaking of attacking from all sides, why don't we have Mike give an intro?
Thank you, Adam. Spectacular. Like, Adam, did I hire you?
Yes, you did. Actually, don't you remember Mark was like he had you have lunch with me. And then on the way, Mark was walking me out. He goes, I have one question for you. I was like, yeah, shoot. Did Mike ask you any scientific questions during lunch at all? And I'm thinking about all the times you talked about the Flyers and the Phillies. I'm like, yeah, I'm sure there had to be at least one question in there.
Now, Adam and I go back. I mean, like you and I mean, unbelievable.
2004.
So we're like totally dating ourselves, and like I'm listening to all of us. Like, poor Brian doesn't. He doesn't have this. Like Barry and Hillary and I. Dear God, all the years of connections. So it's funny. Yeah, no, like I grew up at Centocor pre J&J. I had that blessing. Remicade, like that was like the first thing I ever worked on, and then the blessing of working on Symphony and Stellara and Trimphia. I mean, it was like tremendous to have a opportunity to play with all those molecules, which is unbelievable. Like not too many people. I mean, think about it. You guys in particular. How do you have like in the drug discovery business? Like this is not something normal where you get to have your hands on so many molecules that actually went into people and made an impact. So that's something special.
It's pretty incredible. Not only that, you had your hands literally on the paper
On them.
copies of the vectors,
Yeah.
right? In the binders that were in the lab.
It was unbelievable. So that was fun. Adam, thank you for bringing that back. That's exactly right. That's incredible. But then, like the times were changing, and it was time to move on and go to what was, I think we all thought was the next thing, which was cell therapy, which I think it's still the next thing. And that was, I think, one of the points of our first podcast, which was, look, this is the state we're in. Like, I think we think there are, the cell therapy business is, it's just like the antibody business, which was, anybody who's a little longer in the tooth knows this stuff, which is, look, it's ups and downs, and, it's going to go forward. Things to work out, things to understand. And that's where, and, the conversation earlier with regards to the news regarding, in vivo cell engineering, that's like, I'm a huge fan of that. And we'll see where that goes. But there's details to be worked out, and we'll see where that goes. But it's been a pleasure. Adam, oh, dear God. He we grew up together, like
Sure.
I don't think I'm exactly his father, but we
I have a feeling my personality would have turned out a lot differently.
We grew up together watching that stuff and doing things together, and I can still visualize Adam in the lab. Unbelievable. The music he used to listen to.
Still do.
So much fun.
Heavy metal, man.
And that's probably a good segue then into the complexity of flow cytometry growing up together with cell therapy. So I'll try to, I guess, transition back a little bit to the paper from memory lane here. So, with this paper, the reason that I selected this one, as Adam said, it is around one of the, or actually the oldest commercial cell therapy in existence. And I really liked that they were just very detailed in exactly how they were validating. They talked about how they're using this, in the clinic actually, and then also looking at different instruments and seeing how sort of robust and translatable it is to different sites.
And of course, you had to love it. I mean, they're really talking about all the stuff that we do all the time, right? They did start, you got 2 panels specifically in here. So they started out with a very simple, you got your live dead, you've got your 7-AAD, CD3, CD45, and then their CAR reagent, which is off the shelf, right? It's just it's a CD19 antigen that then binds to CD19. And so, very simple but elegant in the way that you're looking at the presence of the CAR, because the CAR, it has an anti-CD19 arm, right, for targeting to the B cells, go to the B cells, the tumor cells, right, that overexpress the CD19 and then kill the tumor cells. And so that's why you can use the CD19 as an antigen to bind to that arm and detect these CARs. And then they came in with a secondary analysis trying to look at central memory T cells, effector memory, just to be able to distinguish what the types of populations you're seeing with these CAR cells. And I thought it was very interesting too that they noted that these CAR cells can look very monocytic, right? When they're activated, they're larger. And so talking about the gating and how to make sure that you're appropriately evaluating the numbers. And then, I'll let somebody else speak. The other thing I really liked was when they were talking about the lower limit of quantitation, limit of detection, and limit of blank, realistically looking at how many events. And you're talking about the detection being down to 13 CAR events. But needing somewhere in the order of 26,000 actual T cell events to be able to get to that. And they started out with only 200 microliters a sample because we're talking about pediatric samples. So something that you have to take into consideration for your assay when you're trying to set this up.
Adam, I was curious. Reading the paper, that you brought up that CD19 reagent. Mm-hmm. Something we had a little bit of an experience with, and it was definitely different from an antibody-based reagent, mm-hmm. Really in terms of consistency of product quality and having to be a little bit more careful with it. Is that something I was curious? Have you guys used many of these sort of antigen reagents versus an antibody at your company, and do you kind of have to change the way you go about validating it if so?
So I would say the vast majority of things are still antibody related. Specifically, there may be some sort of engineered target on the surface of the cell that would allow for detection with an antibody reagent. Essentially, though, you are treating it very similarly to an antibody in the way that you're performing the various test scripts that they have here and making sure that your assay is locked down. Brian, any thoughts there in addition?
Yeah, and I'll add on to the complexity of using an antigen type reagent. And here as well, we're using a secondary antibody. And I would say that's also relatively rare. In general, we prefer primary antibodies where at all possible. It just takes out one of those extra levels of variability, right? How well does the secondary bind to the primary? Right. So they're starting out with, a relatively difficult thing that has multiple pieces that could go wrong during the staining procedure.
Yeah, no, that's a good point.
Yeah, I just know that I think more recently, some of these companies have come out with a sort of anti-id type reagent that I'm wondering for the CD19 field would be more widely adopted just because it's a little bit more similar to what people are used to.
Mm-hmm. And so one of the things that I really found interesting here was the prevalence of where you've had B cell aplasia, right? So the loss of the B cells, but then you saw the B cells coming back over time, but you still saw persistence of the CAR therapy there. And was interested in the perspective, Mike, Barry, Hillary, in there, your experience there and thoughts there.
I was actually curious. I mean, I know there's very little you can do with 13 events, but I was curious as to if they've tried to further phenotype the cells in looking at exhaustion, because and some markers that might actually be telling of why you still have CAR Ts present. However, you still have B cells also. So I was just I don't know. It kind of struck me that they didn't go further with it, or perhaps they did, and they just were getting inconclusive results because of such a small number of CAR T cells.
Yeah, I felt like they did mention that some of these longer persisting ones that they tended to be higher in naive subtypes.
Yep.
Yeah.
Which was interesting. I think it's funny when you guys brought up this sort of in vivo type of therapies that are coming. I think it's really interesting to look at these subtypes and what comes out. And when I read that in that paper, I was like, oh, that's interesting. Is that unique to Kymriah? I know that I think that's got a 4-1BB in the CAR.
Mm-hmm.
Does the makeup of the CAR change? What persists and what comes out, or is it just these sort of naive cells? They just are longer lasting, and that's what you would get, for these long persisting cells. So yeah, it's a really interesting aspect, and I'm diving more into these subtypes that tend to for these different programs and what lasts and what doesn't.
And this is I think this is the future of the conversation that's going to go forward now, especially with these in vivo approaches where, um, the like you hinted on, Adam, uh, you're depleting these B cells for maybe ever, right? I mean, that's probably what's going on. And beyond the B cell target phenomenon, which is therapeutically like spectacular, but how do you go beyond that? This is going to be the questions, and these are where I think the focus is going to be, which is okay. So are we just going to destroy these target cells forever, or are there other ways? I think to modulate this targeted therapy. So we'll have to see. I mean, like this is the future of these types of therapies, I think. So it should be fun. And I think there's going to be a tremendous amount of new things coming out, and should be exciting to watch. But it's early days, and I think that's the excitement from a dorky, geeky science thing. Um, but yeah, no, this is where the future is going to go.
Yeah, and the thing I found really interesting was that repeat infusion didn't necessarily improve the outcome, right? There was only one patient essentially that showed a durable CAR persistence after the repeat infusion. And so they noted that what you'd want to be able to do is look at functional persistence versus existence. I was wondering how they were going to determine that, but like term it as existence was a cool way to put it. And then talking about how do you reactivate dormant CAR. So to Hillary's point there, and they had mentioned about possibly utilizing some sort of vaccine or a gene therapy type thing to reactivate. And so Mike, to your point, a lot of things that need to be figured out still.
No, I think that you know, getting the T cells activated and going are a component of all of this. Obviously, big
Hmm.
players in the immune system. But one of the things that the three of us have been talking about is there's more cell types there,
Yeah.
and that's probably more important than just the T cells or whatever. And so that's our angle on things, and so we're looking forward to that.
And so I'm curious, like we've in the industry, right? There's T cells, but then to your point, there are NK cells, there are macrophages, right? My own personal favorites there. Talk a little bit about the different cell types there.
Well, I mean, I love Barry and Hillary because they're the experts. I'm just a pretty face, but yeah, um, you think about solid tumors or a tumor that is established in a tissue. Um, yeah, there's players there, and
Yeah.
it's not just T cells. And I think we all feel pretty confident that you got to get them going,
Yep.
and you have to get them reinvigorated. You have to change them. Uh, we use the words alter, we use the words transform, and those are the cell types that it's going to be a big deal. That's where you got to. That's where I would put my bets. That's for sure.
Yeah, as Mike was saying, I think the traditional CAR Ts going into T cells have had a lot of success in heme malignancies, but much less so with solid tumor. And as Mike kind of hinted at and mentioned, we think, and obviously other people think that combating that sort of tumor suppressive environment that you get in these solid tumors is going to take these other types of T cells, or other types of cells, macrophages, as you mentioned, and K cells, and in concert working together is the way that we're going to be able to tackle that sort of tumor suppressive environment that you get with solid tumors.
So you see more of an interplay of the different types rather than just one cell type.
Exactly. And then maybe even, other non-immune cell in that environment, we may need to go after those type of cells too, to modify them and get them involved to combat that. Some of these sort of other supportive cells may need to be part of the solution.
I think everybody's got the right idea, which is, all right, there's a T cell we got to get going. We have to get a macrophage going. There's probably other cells people wish they could influence. Um, that's the mindset. Get all those cells thinking and reacting differently. That's the key. And the question is, well, how are you going to do that? Like that's the big. I mean, Hillary knows this better than anybody. I mean, it's like she's clearly, she watches it
Do
from a
I?
tissue perspective, and she knows this. She watches things happen in vivo. But that's the key. I think. You got to get everybody going. It's not a I put a post on LinkedIn where you know cancer in particular, maybe all disease, is a disease of tissues. Everybody interacting, all those cell types interacting um, differently for whatever reason. And this is the cross we have to bear.
So, Mike, if I'm hearing you correctly, and apologize for completely nerding out, but we've got individual lions, but what we need is the Voltron robot to come together and have all these cell types in one.
Yes,
Yes.
I don't know what that reference is exactly, but
Oh
yes.
no! Come on, somebody else back me up
Oh,
here.
no, I got you, Adam.
I got you, Adam.
Towards your point and to the paper and things like this, I guess we'll have to go from a 4-color phenotyping paper, out to like at least a 16 color to get all these different cell types there and quantified, and who knows how many more, I suppose. But
45. Let's just do
Yeah.
that. Come
All
on.
right.
Go big or go home.
That's it.
Yeah, no, I mean, like that yeah, it's I don't know. I've never been a big fan of well, maybe this is inappropriate, but the cell types and the like they're changing all the time. And like this is what we are. We are complicated biological entities. And yeah, no, it changes every single second. And that's what I think is driving the cancer phenomenon. And yeah, look. You guys are the experts. Tell us what's going on.
Well, I think another point is, oncology therapeutics have always been so focused on targeting a singular thing, and that we know that doesn't work.
Right.
The combinations, the time you have to, be elegant with the timing of when you do combinations based on the cellular changes. And I think between what you were talking about in the intro with the in vivo delivery of mRNA and targeting different immune cells and also the stroma, like the epithelial cells and some of the things that are produced by the epithelium to actually get immune cells going. I think that could be a really nice way to target, using the in vivo mRNA expression on either immune cells or just by targeting genes in the stroma. That could be a really nice way to combat cancer without having to dose patients full of tons of chemos. And it could be a really elegant system. But yeah, it's the stromal compartment and all the immune cells involved, especially in solid tumor, like Mike said, is changing constantly. And by going after singular targets, we're not going to get anywhere.
Okay. Combinations are of course difficult within the pharmaceutical industry, right? Like, if you're having just one thing, it's fine to test because you've got all the IP associated with that. But then once you have multiple, then you've got to ideally test like the best of both combinations. And it starts to add up quickly in terms of the clinical trials and things like that. But I agree that this is a complex thing that we're going after. So there's not usually a simple answer. Sort of like with antibiotics, there's never just a single antibiotic that will solve all the problems. It's got to be a cocktail of antibiotics that's going to prevent a large portion of the bacteria and keep them from mutating away from where you've tried to drug them, exactly like cancer. So yeah, it makes sense and means that we'll constantly have to spar and look at different ways of assessing both what the immune system looks like and what it's doing on an active basis. So exciting times moving forward.
Definitely.
Yeah. In terms of this paper, anything else, Adam, that we wanted to call out?
Really just one of the points mentioned at the outset, they were limited by the amount of blood that they could use because it was pediatric. They were also using the same sample for PCR that they were using for the flow analysis. And so it is very important to absolutely define your LOD, LOB. But the more blood that you can use, the better off you be because you have more events and your accuracy increases. Now, one of the things that they had recommended because of those very low numbers that starting the low volume of blood was that they recommended using any value between LLOQ and LOD, so the lower limit of quantitation and the limit of detection, just as a positive, not as an absolute, with the reason being that you're talking about so few events, right? But as you go further higher, you have more certainty in the determination of the absolute value. And so it is really important to keep in mind the input event input there. The other thing was stability, right? And they had seen that with CAR, there was a significant drop off stability within the first day, even. However, as you got further out, you were still able to determine patterns. And so it still was informative to assay beyond that first day. Again, what we talk about with clients all the time, the sample matrix and the shipping and getting things to us first overnight is always important.
I found that interesting as well, and they were looking at both the sample stability after they did the blood draw, but then also the sample stability after they did the T cell infusion there. So yeah, that initial drop has been reported and makes sense in terms of a lot of the cells just don't make it. So okay, excellent. And Barry, Mike, and Hillary, any other sort of last thoughts on the paper itself?
I don't think so. I think kind of brought up what struck me.
Yeah, same. I'm not a flow expert, so I don't have any additional comments on the paper.
Okay. Well, in that case, I guess we can move right along to the fan feedback request for this month. And this month's request is, what is your favorite automated gating software and why?
Oh my God.
So you're talking about nerdy questions there. This is pretty deep in the weeds there, right? So for all the Elastigate fans versus the Flowsum fans, want to hear which hill you want to die on and why.
Flow AI, and then I'm sure that we go to Cyto, we're going to see even more. It's expanding every day, so always more tools and more toys to play with.
I don't know, Barry, Hillary.
I would be more interested to hear what you guys think is the best.
Fair, and ideally we'll get a couple responses from fans to let us know. We'll read on both Spotify and Apple Music when people get back to us with comments. So that'll be exciting to look forward to. Adam, any thoughts on yours?
No, it's really good, though, that you're looking at it in a positive way, because I'm sure if you spun it the other way, you actually might get more responses. What is your least favorite automated gating platform? Then all of a sudden, I'm sure people will be coming out of the woodwork to tell you. And so, I mean, to be fair, we're in the process of evaluating various different automated gating, and you've already brought up a couple. It's just one of those tools that you got to have now because of the larger data sets, and it's not feasible to really just do hand gating all the time on these gigantic panels and be able to rapidly turn around data sets in the time that clients need them. And so you do need a component of automated gating there.
Yeah. Indeed.
So automated gating is like a real thing?
Yeah.
Oh, yeah.
All right. I'm just totally ignorant. Barry, help me here.
No, I mean, it was something. It's funny because at our previous job, we really were looking at it just because we were interested in collating larger data sets, right? So flow data with functional data, with growth data, and having something that across groups, across individuals, that we felt comfortable we could combine these data sets and have them be meaningful is really key.
Yeah, so Mike, just as a little bit of a background, with a lot of these, they're AI based, and so or machine learning based, where you input the gating philosophy, the gating hierarchy that you've got set, and then what it does is it utilizes that as you input samples to be able to automate the gating. It's good, right? But you still need somebody on the back end to just go over, make sure that the gates are where you would expect. And especially too, when you've got unique samples, something like, ALL, or one of more heterogeneous types of cancers, you would want to make sure you're checking it over because the phenotype can change somewhat enough that it could confuse the machine learning there.
So, are you referencing my age and my inability to comprehend those complexities?
What I'm wondering is, do you have ChatGPT on your phone?
I don't have nothing. I don't know what they're
Or
talking
have
about.
you gotten past a flip phone at this point in time, or you got a smartphone? What do you got?
Oh, my daughters. Oh, it's unbelievable. That's good. No, it's good. So essentially, like, you can, like, you know, plug stuff in and push out the data and get a interpretation.
Hmm.
That's what I'm hearing.
Yeah.
Spectacular.
with caveats, right? And that's what we're looking for from the fans that would comment on the podcast to let us know what sort of caveats that they usually put into it. Definitely everything that we're looking at, we're looking at making sure that there's a human in the loop, which is we think critical to making sure that we're getting intelligible results overall.
Yeah.
But absolutely, for doing the initial gating and doing then these automated gating solutions can take a lot of that away and get you, as Barry mentioned, a lot more data with a lot more speed there.
But the amazing thing is too, first when I started at the company, 8 years ago now, people were loath to use any automated gating, right? There was no trust in it, and now you're seeing posters on it at Cyto. When I went up in Montreal a couple of years ago, they were talking about automated gating, and now you really can't do these big data sets without it. And so just the way that the industry is moving.
That's awesome. No, that's great.
Yeah. But then it does bring us into the ending portions of our podcast here, and very related because with those automated gatings, we're really looking through a large amount of data to pull out the important pieces. And likewise, with Easter coming up, I wanted to ask our guests, what is the most difficult Easter egg or scavenger hunt that you have ever either designed or participated in?
The one, the one when my mom forgot to boil the eggs and used real eggs instead.
Oh no!
Oh no! Hopefully, you were very effective at finding the eggs that year.
That taught her not to have a bunch of 5 and 8-year-olds chasing around Easter eggs that were real, or sorry, not boiled. But yeah, it was pretty awful. So that was quite literally difficult.
Oh, that must have been a mess.
Yeah,
Barry, you're a heathen. So what?
Nice.
What's that going to be?
So it was actually interesting. So we, when my kids were young, my stepsister worked at the White House, and we got an invitation to the Easter egg hunt on the White House lawn, which everyone was really excited about. But kids had to get all dressed up, which, a couple young boys wasn't so easy. And there was a lot of standing in line. So it seemed like a great idea at the time. And, the pictures looked great, but man, it was.
Who was that, Bush?
That was probably, yeah.
That was Reagan, wasn't it?
Yeah, I think it was, or maybe early Obama. It's in that same, that transition era.
How about you, Mike?
Easter egg stuff. Oh dear God. I don't know. Like nothing dramatic. Probably an event or two where the Easter Bunny needed to be taken down, and that was probably it.
It's good. I will say for mine, I'm not going to go with most difficult or whatnot. I'll just go with family lore here. One of the times we did an Easter egg hunt and my parents put out the eggs, and this was the impetus for us switching over to plastic eggs that had something like money in them.
Yes.
We had, Hillary, we did boil the eggs. However, we went through and nobody could find the last egg. We searched for a couple of hours for this last egg, and it never turned up the whole rest of the year. I kid you not, the story goes that 10 years later, we found the egg. And you can just imagine what it might have been like at that time. And so I don't know how much there is really truth that it's 10, but that's the story that's told. And so, make sure that we tried to do a map of where all the eggs were, or my parents did, and it still didn't really help. I guess, you get late night the night before, the Easter Bunny's coming, and somebody, I don't know, maybe there's some Easter wine, who knows.
What was the most money you put in the egg?
My parents generally the Easter Bunny at my parents' house, let me put it that way, was typically more generous than, at my own house. And so I believe that there were like 20s in there occasionally.
Oh, wow.
And not only that, they had like the ability to swap at the end. And so all the kids had their eggs out, and you could take this one, trade with this one. And it was always fun.
It's unbelievable you had 20s in there.
Well,
like for my
this
kids, I'm still doing like 25 cents and dollar bills. Like
this isn't like recent though. Like with the grandkids, this is not like when I was little. I think it was a quarter. It may have been a nickel. Who knows?
I too am still living 30 years ago with my kids.
How about you, Brian?
Used to take kids geocaching. So we would go out in the woods and like look for random like ammo boxes.
What?
Yeah.
Ammo boxes?
And that's the thing. It wasn't full of ammo, but it's full of like little like toys and pencils and stuff like that. And the ammo box is just weather resistant, right? So that's why they put it out there. And you're supposed to say again?
Where'd you grow up?
I do this in Georgia, but then also geocaching is like national, so you can go anywhere in the U.S. and usually find a couple boxes that are hidden somewhere.
Oh, dear God, that is unbelievable.
Yeah, but you can look on your phone and find a geocache site near you and go for it. There was, I think, 2 or 3 different ones that we went to. One was like inside a hole in a tree, and then the other one was like 10 feet up in the branches of the tree, and we spent something like an hour looking for that one, and then finally somebody looked up and saw it. So very very challenging. But yeah, I've always stuck to the plastic eggs as well for exactly that reason. That's terrifying.
That's awesome.
Yeah. All right, and with that, I think we'll close it out. And thank you all for joining us. For everyone that's listened to the episode so far. Next episode, we'll be talking more about instrument harmonization and what's in an MFI. And again, thank you to our guests for joining us and for talking about cell therapy and future of CAR-Ts here.
Yeah, thank you. Really appreciated Barry, Hillary, Mike, having you guys on. It was an absolute pleasure, and thank you to the audience for listening.
Would love to participate in the MFI conversation. That would be so entertaining.
Thanks for inviting us. It was a lot of fun.
Thank you.
All right, take care.